Thanks for joining us today. This is going to be a tutorial kind of presentation. It's called Retrieving, Cleaning, and Annotation of Genetic Variants from ClinVar. And first, I'm going to introduce the database of use here. ClinVar is a freely accessible public archive, which I'm sure that most of you are aware of. It holds public archive reports of relationships among human variation and phenotypes with supporting evidence. Currently, it holds more than 300,000 genetic variations, from small single nucleotide variants to large-scale copy number variants. Genetic information, it's nearly available for 10,000 phenotypes, including disease traits such as binary traits as well as quantitative traits. Next generation sequencing has allowed the constantly growth of reports of this database. And last year only, the amount of entries nearly doubled from what it was available on 2016, which is really encouraging for this database. Currently, it holds more than 500,000 entries, which, since I already mentioned, only more than 300,000 are valid entries regarding genetic variation. So, how this database works? It's a really comprehensive compendium of that accumulates genetic variation from multiple sources regarding researches, but also clinical laboratories and expert groups also upload information. And the database itself annotates these variants and also connects these reports to external resources, most of them coming from also NCBI resources. We also have the presence of specialized groups that actually curate these entries of the database. And all of this in conjunction is constantly happening. ClinVar gets updated nearly every week. So what are the objectives of this database? First, to determine if a genetic variant is pathogenic, to determine pathogenicity, clinical usefulness, and clinical validity of this genetic variant. So on a personal side, I would say that these objectives are usually accomplished when we are in the presence of other similar entries. If you're submitting something new to ClinVar, you won't get much information about your own variant. Also, the database usually holds spurious reports of variants with no valid information. For example, there are some entries with, where the phenotype is not provided. So you can get pathogenic variants for phenotypes that you don't know. So, the database is quite popular among clinicians, but it's also known for these kind of not well-addressed issues. So, in my personal opinion too, also ClinVar is only getting better. It's constantly improving and updating itself, and the more variants we accumulate, the more accurate we're going to be able to predict pathogenicity of novel variants. So how does it look? I'm sure you're all familiar with the browser. And also, how does it look an example query? This is an example query that it's shown on the paper of ClinVar. And you can see directly that it provides comprehensive information regarding the variant, date of submission, and all kind of annotations and connections to the NCBI resources. But also it's evident that there is an excess of information, and it's difficult to handle, difficult to export, for example, this table to an Excel sheet. And it's not directly scalable to a genome-wide level. For example, if you had some genetic candidates, it will be hard work to keep browsing and browsing for different entries. Also, the entries, as we can see here, are kind of awkwardly annotated. I know that this notation is systematic, and you can actually determine which type of variant is this. it's not straightforward and it's hard to read. And here I provide a couple of more examples of this kind of entries. So besides the objectives described by their own database, we usually ask additional questions that require the full database. For example, is the disease associated to a certain type of genes, or how these genes behave in the general population? And to answer these questions, the database, as it stands, it does not allow us to answer these questions. So ClinVar provides on their FTP site a complete BCF file. This is the variant calling format that contains the full amount of information of the complete database. This file potentially can allow us to answer these questions, but it also requires external resources to properly handle these files. It's difficult to modify it without any bioinformatic knowledge, since the database is also focused on clinicians' use, and it also has excess of information. So for this tutorial today, I will show you how to retrieve all ClinVar data in a friendly way, how to generate a reliable set of genetic variants for downstream analysis, and also how to get a disease or gene-oriented reports from the database. And finally, how to add or connect ClinVar with other sources of information. This is commonly known as annotation of a ClinVar report. Initially, this tutorial was designed to show you step by step how you could do this. But at the end, I thought it was more useful to actually create something that will allow you to do this automatically. So that's the origin of this PER program called ClinVar Viewer, which I will show you now. So first, you need the complete ClinVar data, and this is obtained in the FTP site. It's called on the tab delimited side. You can see here that you have the BCF entries, but I strongly suggest to get the tab delimited file, which is also weekly updated. Inside this folder, you can get the variant summary, compressed, of course, and this weights uncompressed around 200 megabases. So this is the complete input file upon which we will work, and we also and we don't need nothing else, just a folder with the program ClinVar Viewer and our variant summary uncompressed. So now I'm going to go to the terminal and show you how this program works. Well, first, this is the run of the program without any parameters. And you can see immediately that the first objective of the program is running now. So we should wait a couple of seconds. So yes, now the program is actually running. And what it did on the background just now is to create a variant summary .clean, which extracts the core information of the variant summary file and creates a smaller file easy to read. This variant clean looks like this. So first, I'm going to show you how the bulk database looks. So these are around 30 fields, so it's also really hard to read. But when we move to the just created variant clean analysis, we can immediately detect which type of variant are we talking about. So now we have genes, type of variants, and molecular consequence. Here I would like to come back to the presentation because the molecular consequence is not directly seen nor in the BCS file or in the variant table 2, in the initial variant summary. You cannot extract or detect immediately which variants are missense, stop gained columns, or that kind of molecular consequence annotation. This is made indirectly by the program, and this is the variants as they stand initially, and we can now annotate which of these variants are missense, synonymous, or splice donors, or even non-coding, using the sequence variant nomenclature. So the program grabs all of these rules, which are quite complex, and integrates into the downstream analysis to generate the variant clean report. So now we can see what we have. We have three options: header, summary, and annotation. So the variant.clean has this structure, only 12 fields in comparison to the nearly 30 that had the ClinVar tab delimited file. And now we can see what we have here. So we can just type summary, and the .clean generates a summary output containing the main features of the complete database. So now here we can see in decreasing order the type of variants that are contained in the ClinVar database, the consequences that are inside, and also the clinical significance of these type of files. So when it gets interesting is when we use the summary file in a gene-wise or disease-wise report. So we can do the following. We can just request a summary, but of a gene, and in this case, let's try with SCN1A. You all have your favorite genes, so if you type this kind of inputs, you will get a report that it's gene-wise. So now you have a summary only for the gene SCN1A and what is available on ClinVar on the entire database regarding this gene. So we have 6 types of variants with 9 types of consequences. Among them, 632 missense variants. And here, when you go to the gene-wise report, you can get, well, of course, the amount of genes is 1. You can get all the diseases that has been associated this gene. You can try it with another one, for example, green 2A. And you get immediately an independent report of this gene. Of course, you can also try with phenotypes. For example, you can type a summary of this. This will be a keyword, and you can type epilepsy. Oh, that didn't work. Let me try this one. No, that didn't work either. I'm sorry. But, well, on the background, this program creates immediately a gene summary report where you can see all the gene variants associated to green 2A, or also to SCN1A. So I will try again, and this was working very well. Yeah, there it is. So you get the same report this time regarding a disease. So you have 6 types of variants associated to epilepsy with 12 types of consequences. Most of them are missense. And now we have correctly spelled the epilepsy report of the entire ClinVar database. And you see that here we only have the information that we want. And also, we have already structured the amino acid position and the change independently annotated. So you can just grab this gene-wise report and export it to upload the entire sequence of the protein, for example. At the end, you have all the diseases associated and a key that denotes the specific position of each mutation. That is chromosome, starting point genomic, endpoint, reference allele, and alternative allele. This last field is used for annotation. So this is what we're going to do now. And for annotation, you need only an input with the same key. That is, for example, if we would like to annotate the frequency of genomic variants, we'll need a file like this with chromosome, start and endpoints genomic with the reference allele and the alternative allele. This file, freaknomad.source, I generated myself. You can do it with anything that you want to annotate. And you can just upload it to the program like this. So you say this time, well, we have the option 3 now. We have annotate, and then you put the name of the file, which in this case is frequency of genomic variants. So you just press enter. And it's currently performing and searching each of the variants in ClinVar if they have some frequency reported in control population, such as Nomad database. So yeah, now that the annotation is successful, we can just see how the files are updated. And this is, again, seems like this. We now have an additional field, which is called frequency of the nomad. And then you can just summarize whatever you want. You can do a new summary of SCN2A this time. And now we'll have the complete annotation of SCN2A plus the nomad frequency of all the variants reported for this gene. Again, on the background, a file specific for this is generated, which I don't see right now. Oh, there it is. But yeah, so this is the usage that the program has. It's really simple, but it's also really fast and effective in what we want to do. So, well, as a concluding remark, I would like to keep moving forward this small project to get visualizations of gene-wise report or disease reports, automatic connections to the updates of the database, and also to include more annotations. So on a personal level, I'm happy, of course, to share the tool, and also the feedback is more than welcome. So thanks a lot, and well,